All right, welcome to the weekly seminar GBSBC. Today we have the pleasure to have Dr. Alicia Martin in the room. As a brief introduction, Alicia Martin is an assistant investigator at Massachusetts General Hospital, an assistant professor at Harvard Medical School, and an associate member at the Broad Institute. Her research as a population and statistical genetics focus on how human history influenced global genetics and phenotypic diversity. Dr. Martin addressed the Eurocentric bias in genetic studies, ensuring that genetic technologies do not worsen health disparities. She developed statistical methods, community resources, and research capacity for multi-ancestry studies, particularly in underrepresented populations. Today, we have the pleasure to hear her talk about her project called Genomics for the World, Ancestral Diversity Enhanced Genomic Discovery, Resolution, Interpretation, and Contextualization. Ladies and gentlemen, Dr. Alicia Martin.
All right. Thank you so much for that lovely introduction. I really appreciate it. And thank you so much for being such welcoming and accommodating folks. It's always a pleasure to be here in beautiful La Jolla and UC San Diego. Okay, so I'm going to, sorry, just make sure I can see my notes. All right, so I'm going to talk to you today about a lot of things that are going on in my group. So aside from the introduction and some key takeaways that I'll introduce, all of the work that I'll talk about today is from our lab that's not published yet. So you're going to see sort of a lot of cutting edge work and some projects that are on their way to good homes. And then you'll see lots of preprint links throughout. But that being said, if you have any feedback, I'm really eager to hear from you, so please get in touch. Okay, with that, let's jump in to the link. I think it says we can't see slides.
Yeah, it's not quite. Are you sharing?
It says screen sharing is paused.
Maybe stop share and start again.
Sure.
Do that one. How does it look like? That's good.
Okay. Hopefully you can all see it now.
Yes.
Great. So I'm going to jump right into the introduction then. All right, so as a population and statistical geneticist, I trained in human evolution, human migration history, that sort of thing. So I like to think through an evolutionary lens. That means to think through this lens, we need to start in Africa, where our species originated. Then our species migrated, a subset of our species migrated out into the Middle East, into Eurasia, and subsequently into the Americas. And along these migration paths, our species took a subset of genetic variation with them. So that means that the average African population has about 1 million more genetic variants on average than the out of Africa population. That being said, I want to emphasize that there is more variation within than between populations. So this is a sort of important human evolutionary history that's important to consider when we're designing genetic studies. I think with all of this being said, it's a really amazing time to be working in human genetics. This figure is now about a half a decade old, but you can see that there's just been explosive progress up until this point, and it's only continued forward to present day. Because our genetic studies have just gotten so massive, you're seeing many, many studies that are coming out with the order of millions of participants in these genetic studies. So we've learned a ton about human history, but also importantly about the biomedical basis of disease over the course of these past decades. With that being said, genomics has a huge diversity problem. So this is an issue because if we want to fundamentally understand the genetic basis of disease, we need genetic variation. That's our key ingredient. But if we have less genetic variation because we have a biased snapshot of that variation, then we're destined to find fewer genetic associations overall. This has been a really important problem for a long time. We are certainly not the first to have identified this problem. There are many investigators that are working to rectify this issue, but I think it's important to consider how deeply ingrained and how institutionalized some of these issues of diversity in genomics and more broadly in medicine are. So a fundamental core question that our group is interested in is how do ancestry study biases in genetics impact the generalizability of our knowledge? With that being said, I think there's a few important grounding points that we need to be on the same page about. So fundamentally, we expect that causal variants are by and large shared across populations. That is to say, if we were using a tool like CRISPR, let's say, and to take one variant into one genetic background, the same variant into another genetic background, we would fundamentally expect that its impact on phenotypic variation should be the same regardless of your ancestral background. That being said, like I said with that map that I was showing earlier depicting human history, this history has an impact on allele frequencies across the world, also how correlated those alleles are across the genome, how different environment impacts phenotypic trait variation in concert with genetics. And there's many other complicated factors that weigh into phenotypic trait variation as well. So several years ago, we showed that the impact of this Eurocentric study bias was that there are vast differences in how accurately we're able to predict traits using genetic data, for example, using a polygenic score across populations. So for example, in this plot, I'm showing you a polygenic score that we've generated for dozens of different traits in the UK Biobank data, where we've trained on European ancestry data. And then we've predicted in UK Biobank, but in a range of different ancestries. And as a consequence of these vast Eurocentric study biases, what we see is that there's huge differences in prediction accuracy. So for example, we're predicting about twice as accurately in European ancestry populations as in East Asian ancestry populations, and about 4 to 5 times as accurately as in African ancestry populations. This is not because these traits differ, it's simply because of these study biases. So this is really important because there's also emerging evidence that these polygenic scores and many other facets of genetics are going to depend on these study biases. So polygenic scores are coming to the clinic, whether we think they're ready or not. For example, the eMERGE consortium is working on returning polygenic scores for 10 conditions that are common in the population to 25,000 patients already. That means in the coming decade or so, we're going to see this increasingly in medicine. And this issue is going to impact interpretations related to accuracy across these groups. There's also an enormous amount of promise that we can gain from diversifying our genomic studies. So there's huge scientific opportunities that we can gain from diversifying these studies. So one is novel associations, new biology that we wouldn't identify from variants that are missing in European ancestry populations that are present in underrepresented groups. As a clear example of this, we see, for example, new CRISPR technologies that are treating sickle cell anemia that wouldn't have been discovered if you hadn't done these studies in African ancestry groups, for example. We have the ability also to conduct fine mapping and better pinpoint causal variants as a consequence of leveraging this increased diversity across diverse populations. I already mentioned genetic risk prediction accuracy, and then the last is a really complicated one, understanding how the genome and environment interact together to produce complex trait variation. We're a little lost if we don't understand the range of that environmental variation and study diverse populations that are experiencing all of those types of variation as well. However, getting to this sort of really idealistic scenario in which we have very diverse genetic studies at massive scales for everybody still needs quite a few pieces of infrastructure in place. So for example, we need new statistical methods. Right now, we assume a lot of homogeneity in our statistical methods. And so relaxing those constraints and relaxing those assumptions requires methods development to be able to consider and account for variation across different populations. We need better data and community resources. This is a topic area that we spend a lot of time focusing on, trying to build resources and build new data analyses across diverse populations to ensure and allow new insights from diverse populations. And then research capacity is also very important. So for example, if we look at the representation of diversity in genomic studies, we see that this is far out of step with the global population. But it's not just far out of step with the global population, it's also far out of step from the US population. And just to call out, the NIH is one of the largest biomedical funders of research in the world. So the fact that we're not representing that needs a closer look. What we are doing a pretty good job representing are those NIH-funded investigators who are doing a lot of this work. So if we're hoping to change this picture and to diversify representation at all levels, it also requires heavy investment in training the next generation of leaders in these diverse genomic studies. Okay, so with all of that said, I'm going to dive into a couple of different areas. So all of the research topics that I mentioned will be unpublished work and active projects underway. The first is going to focus on biobank scale studies across traits and ancestries, working on how they empower genetic architecture studies and offer new biological discoveries. So one of the most valuable resources in our field for human genetic studies of complex trait variation is the UK Biobank, which is not a very diverse resource. It's about 95% European ancestry. And so by and large, when people are making use of this resource, they're pretty much doing GWAS, or at least they were when we started this project, in the European ancestry component and excluding the non-European ancestry participants. So even though the UK Biobank is 95% European, roughly, ancestry, that still leaves out 20,000 individuals with primarily ancestries from other groups. And while these ancestries comprise only a small fraction of the UK Biobank, this large resource often means that these are the largest GWAS with non-European ancestries available for many different traits and phenotypes that we're interested in studying. So we ran GWAS across 16,000 different ancestry trait pairs. This included 7,000 different phenotypes across 6 different ancestry groups. We released 12 terabytes worth of summary statistics that you can access, and then you've been able to access for the past couple of years. We also produced a frequently asked questions sort of list of why we did this study, describing some risks, benefits, and limitations of producing this resource. Compared to other UK Biobank-wide kind of resources, we approached this using linear mixed models, which wasn't sort of standard at the time. So this resource also is using kind of some of the latest and greatest tools for conducting GWAS. In this plot on the left, I'm showing you how some of these studies that are, or some of the associations that are being identified as a consequence of doing the multi-ancestry analysis on the top left part of this sort of QQ-like plot, where the x-axis is showing you the minus log 10 p-values of many different GWAS in European ancestries, and the y-axis is showing you what happens when you do the multi-ancestry meta-analysis. So things in the top left are generally showing you things that you wouldn't necessarily discover using the European ancestry groups only. So overall, we found tens of thousands of independent genome-wide significant associations. For reference, the GWAS catalog has about 600,000 genome-wide significant associations in total. So the UK Biobank is contributing a huge portion of these. If we look at how many of these are novel according to by rough terms, so this is sort of a unique phenotype definition, we're finding about 34,000 unique associations. And if you sort of filter down by like kind of broad category of traits, you're finding 5,000 novel independent associations. Then if you filter down to what has been discovered already previously from UK Biobank and exclude those, you're still finding 3,000 novel associations that weren't identified using the European ancestry analysis alone. But some of these were close to significance, including these 3,092. And then there's a lot that are European ancestry enriched as well. There's a couple hundred that bear looking into as a consequence of their ancestry enriched allele frequencies. The UK Biobank is only kind of a starting point. As we all know, the All of Us resource is becoming increasingly powerful and is a really fantastic resource for especially diversifying progress in genomics. So we wanted to understand polygenic score generalizability. And then the UK Biobank, even in the pan-UK Biobank analysis, the like kind of takeaway is that the picture still looks very similar to what I showed with the disparities figure to start with. Like we're not gaining a whole lot in polygenic prediction accuracy, even with the pan-UK Biobank analysis. So we wanted to analyze the UK Biobank data with all of us to try to understand how polygenic risk score generalizability changes. So to do this, Kristen and Ying, a talented postdoc and PhD student in the group, harmonized phenotypes fairly painstakingly across UK Biobank and all of us. Then they ran single ancestry GWAS within all of us and used the pan-UK Biobank GWAS from UK Biobank. They combined these into multi-ancestry, multi-biobank as well, meta-analyses. So there's a lot of work just in the harmonization piece. After all of this, they then conducted and ran polygenic score analyses. And then we held out a subset of individuals in all of us to allow us to understand how polygenic risk scores were translating within and across diverse ancestry groups in all of us, with a holdout set of about 5,000 individuals per ancestry group for admixed American or Hispanic and Latino populations, African ancestry groups, as well as European ancestry groups. There was a smaller subset of Central South Asian and East Asian ancestry groups, so we predicted there as well, but for the sake of sample size, we weren't able to do too much there. And then we also looked at individual level accuracy instead of binning by groups. So one thing that has emerged from our study as well as other studies is that the best strategy for developing polygenic scores across ancestry groups is really dependent on the study. So is this emerging from these biobanks with hospital-based ascertainment, or is it emerging from volunteer-based studies, and how closely does that mirror the ascertainment of the cohort you're trying to predict in? It's also dependent on ancestry. So where did you do the study? Who comprised the discovery cohort? And then it's also dependent on trait and genetic architecture. So looking at these plots, this is kind of busy. So I'm just going to walk you through this a little carefully. So on the left part, these sort of blue blobs are showing you prediction in the European ancestry group in all of us. The middle is showing you predicting in African ancestry groups in all of us. And the right side is showing you predicting in admixed American groups in all of us. The discovery GWAS that we're using to train these polygenic score models is on the y-axis. So you can see the sort of top set of 4 rows is showing you training with all of us discovery GWAS. The sort of middle peachish, I guess, rows are showing you training with UK Biobank data only. And then the bottom row of these heat maps are showing you training with multi-ancestry meta-analysis that combines data across UK Biobank and all of us. So there's some thinking in the field that the biggest is best. And so combining all of the data that you have should perform best. If that was the case, you would see boxes around everything on the bottom row for all of these different traits that we're predicting in. That's not what we see. And in fact, we can kind of extract some trends here depending on where we're predicting and which ancestry group we're predicting in. So if we're predicting in European ancestry groups, we tend to see that forgetting about the all of us data is performing best. So just using the UK Biobank is helping us predict accuracy the most. One thing that we've dug into and kind of learned about this is that heritability estimates tend to be higher for quantitative trait variation in UK Biobank, and they tend to be similar or a little bit higher for binary traits or disease phenotypes in all of us. So this is potentially contextualizing and helping explain why we're seeing some context-specific differences across these prediction groups. In the middle, in the African ancestry target population, you tend to see that for really polygenic things like BMI and height, you are predicting best with a combination of biobanks, for example. But for a number of different blood panel traits, including things like white blood cell count, for example, and hemoglobin concentration, you're doing best by forgetting about the UK Biobank data and just using the all of us data, in fact. The answer is somewhere in between for the admixed American groups, and there the general tendency is that meta-analysis mostly across the biobanks is tending to improve your power for prediction a little bit more. So all of this is to say, this is complicated, and combining all of your data is not always the best strategy, and you might want to think a little bit more carefully about where your data is coming from, how you set up the study design in the first place, or somebody else set up the study design, and what your ultimate goal is with these predictors. Now, in trying to dig in and understand some of this complexity, we dug into a couple of different traits and tried to understand what was going on when we predicted using European ancestry data alone versus when we used the meta-analysis across biobanks or across ancestries within all of us. And in that scenario, in that scenario, we saw, for example, that in BMI, we're largely finding aligned effect sizes and similar prediction statistics within or across different ancestry groups. On the bottom right part of this QQ-like plot, we're looking at a different trait, neutrophil count. And in this scenario, if we use European ancestry data alone versus if we use a combined meta-analysis across ancestries, we can see some big ancestry-enriched effects here. And one of these is a very classical variant, the Duffy null allele, which leads to benign neutropenia. So we didn't discover this. This is very, very, very well characterized, but it has huge implications on how accurately you're able to predict traits across the ancestry spectrum. So for example, if we look at BMI and how accurately we're predicting across the ancestry spectrum in this sort of top panel, this sort of rainbow-y colored panel, you can see that the prediction accuracy using European ancestry data alone with the solid line is basically indistinguishable from how you do at predicting BMI if you have the multi-ancestry data. On the bottom part, though, if you're looking at trying to predict neutrophil count and you have done your study in European ancestry data, you don't have a good pulse on the Duffy null allele here. And you're doing a really bad job predicting across the ancestry spectrum. So all of this is important to account also depending on the trait architecture. Thinking a little bit more deeply about all of us data, I just wanted to highlight some work that's being done by the All by All of Us group. So in collaboration with Konrad Karczewski's group, I don't know why this showed up like this, we have a large number of GWAS that we've conducted across multiple samples and variants. So for example, there are dozens of lab measurements and then hundreds of phi codes that have been analyzed across ancestry groups. The largest number of GWAS has still been conducted in the European ancestry groups because it's the largest sample size. But you can see that the number of phenotypes that have been analyzed in African or African American groups, as well as Admixed American or Latino groups, is also substantial in all of us. And they're also sort of punching above their weight in terms of their discoveries for those underrepresented groups, which makes sense based on study biases. So we can also combine the sequence data from all of us in UK Biobank to increase power for gene burden testing. So in total, we've looked at dozens of lab measurements, phi codes, including 860 across all of these various trait domains that you can see, and then also 10 physical measurements. We've conducted burden testing using loss of function variation, missense variants, and also synonymous variants to sort of maximize power for gene burden testing. When we do this and combine UK Biobank and all of us, what we can see is that there's increased power comparing the x-axes for UK Biobank burden testing versus a meta-analysis across UK Biobank and all of us on the y-axis. In particular, some of the loss of function signal that's coming from the meta-analysis seems to be driven by the fact that there are some higher allele frequencies in the all of us cohort compared to what their frequencies are in GBSBC, which is a UK Biobank-based exome sequencing cohort. So we're just starting to dig into these associations, but we expect that there's novel loss of function variants associated with disease that we're only identifying from the combination of all of us and UK Biobank data. But we are not going to be able to systematically go through these, and this is sort of a call for all of you to try to help us understand the disease biology that we're learning here. So with all of that, I just wanted to mention that the All by All of Us GWAS has been publicly released. So there are full matrices of summary statistics and per phenotype tables that you can use on Google Cloud to try to understand and mine all of these. There's a featured workspace on the All of Us researcher workbench that we've helped develop that also gives you information about these phenotypes, how this data is laid out, and helps you hopefully try to mine this data so that we can try to understand all of this disease biology together that we're learning from this. There's also a public browser of results, the allbyall.researchallofus.org website allows, it's live now, and you can sort of browse through all of these results at your leisure. I don't know, sorry. So I wanted to also mention that these are really well-known biobanks. Many of us have accessed summary statistics from some of these or have worked with some of these data sets. In other parts of the world, there are a lot of new biobanks that have been analyzed to a lesser extent. And in particular, I just wanted to call out that the East Asian colleagues have some large biobanks that they've started banding together and in sort of a federated way, doing large multi-ancestry analyses as well. So for example, a talented PhD student came to the group with a Korean cancer prevention study that had about 160,000 individuals that had biomarker data, that had lab measurements, that had genetic data as well, and was interested in doing some large genome-wide association studies and was interested in understanding genetic correlations among traits, the traits' genetic architectures, doing these kind of basic comparisons and characterizations within and between various studies, and then as well as some fine mapping of some of those top associations that were novel and new in this cohort. So the Korean Cancer Prevention Study, or KCPS2 for short, has over 150,000 individuals. Yeonho has analyzed this data set with another Korean biobank called COGIS, a biobank in Japan called BBJ, a Taiwanese biobank TWB. Together, these make up about 500,000 individuals, which makes up about the sample size of UK biobanks. So there's starting to be some real and exciting progress in banding together all of these biobanks to analyze trait variation. So as some interesting biology that I just want to highlight that's sort of emerging from this, these biobanks also measure different things. So there was a really high novelty rate in the GWAS that we were learning about from thyroid stimulating hormone, which was measured on all individuals in this biobank, but is not commonly measured biobank-wide across in different studies. There was another measure that is not typical here, carcinoembryonic antigen, and there was a novelty rate of like 62% of the loci were new here. Because of this, we wanted to look further into these novel variants and try to understand their functions. And so here we have a plot of risk allele frequency in KCPS2 and then risk effect size along the y-axis. This sort of smile curve is very characteristic and is kind of shaped by our statistical power to discover these things. All of the novel loci that are identified here are in red, indicating they're new to this study. And one new and exciting locus that I just wanted to point out is this new locus near or in CD36. This is a missense variant that has a much higher frequency in KCPS2. It's at about 7% frequency there. It's at about 3% frequency in a nomad database of East Asians, and it's basically absent, or it is absent, in European ancestry individuals in nomad. This gene also makes a lot of sense with regards to its biology related to thyroid function. So it's involved in fatty acid translocase. This is sort of an exemplifying discovery that is enabled by leveraging samples from diverse ancestry groups and wouldn't be possible from the European GWAS alone. This gene has also been associated with tumorigenesis and development of cancers. So clearly there's some work to be done here, as basically thyroid levels have sort of a trade-off between autoimmune disease on one extreme and cancer predisposition on the other extreme, sort of working towards balancing these risks. Okay, so diving in next, I just wanted to talk about some big work that we've got going on in psychiatric genetics in particular, and how novel insights are being fueled towards empowering severe mental illness studies and trying to understand evolutionary genetics as well. So to achieve larger genetic studies in parts of the world that are underrepresented in psychiatric genomics, which has been overwhelmingly of European descent, we launched this project that we've called PUMAs, the Populations Underrepresented in Mental Illness Association Studies. This by and large focuses on populations in Africa and Latin America, in particular in Eastern and Southern Africa, which are especially poorly represented in genetic studies so far, as well as in Latin America. We launched this initiative, the Stanley Center Global Initiative, where I'm based at the Broad Institute, in 2014. The NeuroGap GWAS, which is taking place in particular in Ethiopia, Kenya, Uganda, and South Africa, in 2015. And our goal was always to collect about 35,000 cases and controls across these countries, which was a pretty lofty goal at the time. Thankfully, at present, we've surpassed these recruitment goals, and all of these participants have been sequenced now. So building on the success of NeuroGap, we also banded together with other colleagues at UCLA and at Rutgers and have bolstered also the sample sizes being collected in Latin America. Altogether, we're building a sample bank of 180,000 participants, cases with severe mental illness, as well as matched controls. We've applied a blended sequencing approach here instead of using standard GWAS arrays, and we're working with colleagues all over the world to analyze these data that helps design these studies and generate these. Trainees are based at all of these institutions that are participating to analyze the severe mental illness heritable basis. So one of the first technical challenges that we wanted to tackle here is in how we design our genetic data generation strategy. So in European ancestry studies, we have a pretty easy and relatively low cost genetic data generation strategy. You can use chips and GWAS arrays to run these at scale with low cost. A challenge with that, though, is the backbones of a lot of these GWAS arrays is also vastly Eurocentric. So to try to get beyond this and to also empower new genetic study analyses, we designed this blended genome exome approach that combines low coverage whole genome sequencing with deep exome sequencing. This is really great because you get not just the common genetic variants that you can impute well, but you also get high coverage coding variants that are enriched for function that we can also interpret. So this provides, rare coding variant capture. It's unbiased common variant capture as opposed to ascertaining on more common variants and allele frequencies in European ancestry groups. And then it also gives us a single shot run through a sequencer. So instead of having to prepare 2 libraries and sequence them separately on the machine, we sequence these all together. So you get definitely the low coverage data and high coverage data all at the same time, which leads to fewer disjoint analyses. And we've deployed this at the Broad Institute for $100 a sample. So this gives us a really cost-effective way to capture the full spectrum of genetic variation. Digging into this a little bit, I just wanted to show you some comparisons in terms of how many genetic variants we're capturing compared to a standard genotyping array, the Illumina so-called global screening array, versus high coverage whole genome sequencing, which is the gold standard if you were to do a large-scale study with no bearing for cost. So by and large, we're capturing the vast majority of coding variants. So if you're looking at this table at the sort of leftmost column, I've separated out by common variant frequencies versus rare variant frequencies. And then this sort of second column is showing you whether this is non-coding or coding. I'm showing you how many variants you're capturing with BGE versus with GSA and whole genome sequencing. So if you're looking at these, I guess, 4th and 5th rows, you can see that we're capturing the vast majority of coding variants that we're seeing in whole genome sequencing. We're capturing the majority of variants that are common in the non-coding regions as well. And we're doing this at about 1/4 the cost of what we do with high coverage whole genome sequencing. It's a little bit more expensive than with a genotyping array, but again, we're trying to go for sort of a Goldilocks approach, not a perfect approach here. So this allows for scale as well as, some new biology that you can look into with these rare coding variants. Okay, so with our sequencing strategy in place, we have been looking more deeply into the neurogap psychosis study. So this is, again, a case control study with over 43,000 Eastern and Southern African populations. This study name is short for the Neuropsychiatric Genetics in African Populations. This is one arm of neurogap. There's others that sort of relate to this. So we have, for example, a capacity building program that goes hand in hand with this research program. We have been looking at sort of diagnostic bins within the neurogap cohort. So this is a study of psychosis. Under that umbrella, most of the participants have schizophrenia, and then right behind that is bipolar. But some of the sites have a little bit more bipolar, for example, in Uganda compared to the others. So Lorato was a Ginger Fellow that was training as an early career researcher, and we worked on some projects together that she was really excited about. And so after the Ginger program, she came to the group as a postdoc to work more in depth on the neurogap data and more closely together. So she's conducted some GWAS in neurogap by disorder. So for example, the schizophrenia GWAS includes over 11,000 cases and 17,000 shared controls with bipolar and psychosis. Bipolar has nearly 6,000 cases, and then the psychosis sort of umbrella definition has about 17,000 cases. This is a Manhattan plot on the left that's showing you what the results look like from the first stage GWAS of the neurogap data. And here what we're seeing is that there's only 2 genome-wide significant loci. One of them is in TMEM150C, and one of them is in PTPRD. These are only significant with the schizophrenia GWAS. So we don't see sort of genome-wide significant loci with the grouped kind of like psychosis definition. So we have to get a little bit more granular in our phenotype definitions. And there's a lot of work underway to look beyond just diagnostic bins, but to also look at symptom level data as well, because these individuals have been phenotyped at a reasonable level of resolution, including things like mania symptoms, depressive symptoms, and so on, as well as substance uses. So PTPRD in particular seems pretty interesting. It bidirectionally induces pre- and postsynaptic differentiation of neurons. It impairs synaptic function and behavior in knockout mice as well, and it's been previously associated with some neurodevelopmental disorders. This one's kind of interesting as well because its frequency is about 5% within neurogap, and it's basically absent everywhere else. And it's particularly enriched within Kenya. So we see an ancestry-enriched variant that clearly has some neuronal function coming up here. We've tried to look at how consistent effects are with the large-scale psychiatric genomics consortium data that's been by and large composed of European ancestry and to a lesser extent East Asian ancestry data. We do see pretty substantial consistency. So perfect consistency would fall along this black diagonal line. We do see some variants that have flipped effects, and we're not totally sure why. Although when we look into those flipped effect variants, we do see that they tend to cluster in the same intergenic region, so more work to be done there, as well as a long non-coding RNA. So we're not sure why those are popping up, but by and large, we do see pretty consistent results with what's been identified with the PGC data. Now, 2 new associations is not totally overwhelming from a cohort of this size. So I think a lot of the value in adding this data to the PGC data is actually going to come in resolving which variants are more likely to be causal given the potential in fine mapping here. These are incredibly ancestrally diverse individuals. They have ancestries coming from many different places. So we think this is going to help us pin down which variants within a given region are causal and which ones are kind of along for the ride in correlation space. We chose to study psychiatric disorders in these populations not because there are more of them, but because of their remarkable diversity, like aiding the fine mapping endeavors, for example. But another huge form of diversity that I'd be remiss not to mention is their ethnolinguistic diversity. So as part of this study design and setup, we designed a demographic survey that allows up to 3 languages to be recorded per participant, their parents, as well as their grandparents, giving us a total of 21 different languages that can be endorsed for each participant. There were in total 130 language options that were given in this survey for individuals, and 90 different distinct languages were reported among these participants. So this is huge ethnolinguistic diversity as well. This is close to what's reported in their parents' generation. So the participants are reporting 90 languages across all, the participants are recording 90 languages. At their parents' generation, we're dropping off a couple. At the grandparents' level, that drops off a couple as well. But notably, the participants are reporting what their grandparents' languages are, and they may not be fully aware of all the languages they're speaking. So that's one sort of caveat. Across sites, we see that there's the most distinct languages being reported in Kemri, which is a coastal site in Kenya, as well as in Uganda. So these are the most linguistically diverse places, but there's a huge amount of linguistic diversity among each of these sites as well. We wanted to measure the relationships between genetics, language, and geography, kind of tracing back to some of the human evolutionary roots. So we measured different types of genetic, geographic, and linguistic types of variation to be able to align all of these into the same coordinate space. So to do this for genetics, we ran principal components analysis using autosomal data as well as X chromosome data to see if there's any sex bias in correlation with language versus geography. For geographic coordinates, we used 2 different metrics. So we used the geographic site where they were recruited from. Participants aren't giving us addresses that we can easily look up at the individual level. So we're taking the sort of site level information. And then we also derived a sort of centroid of non-English languages reported by participants. And by that, I mean, they're reporting to us what languages they're speaking, and we're looking in these database called Foible to understand where is the sort of center of mass for where that language is spoken. We average that using a weighted average across their sort of language tree to kind of pull that information out. Sorry, just to describe the linguistic variation for geography, but the linguistic variation measure that we're looking at is based on how similar language sounds are across participants. So what kinds of consonant sounds and what kinds of verb sounds are you hearing within these languages, and how similar are these languages in comparison? So this allowed us to get a sort of data point for each of these that relates all individuals to each other based on genetic data, geographic data, and linguistic data. So now that we have all of these measures, our next step was to compare the pairwise relationships between genes and geography, language and genetics, as well as language versus geography. And to do this, we used a method called Procrustes analysis. So this method is basically a matrix rotation and scaling method that puts people on the same coordinate system to be able to minimize the distance from one set of matrices to another, providing both summary metrics as well as a visualization of their overlap. So Mary is a super fantastic post-bac turned PhD student in the group, and she looked at these correlations across groups. And what she found is that language and geography have the closest relationships, with a correlation here of about 0.7, which is quite high. This is especially true when using the language-based geographical coordinates. All of these are quite correlated, so these all have a correlation over 0.5. And in general, I just wanted to highlight that this ethno-linguistically diverse region may have other confounders in the GWAS or in genetic studies that could be accounted for with other metrics like language information beyond just geographical information, which is going to be spotty at best in this kind of study. So now diving back in to some other new work, I just wanted to come back to this outline and mention we're very interested in dissecting pleiotropy and delineating genetic and environmental effects, particularly in respiratory diseases, because there's such massive disparities across populations. So respiratory diseases make up a huge burden of global diseases. So COPD is the third leading cause of death globally. Asthma is the most common chronic disease of children. Lung cancer is the leading cancer-related cause of deaths worldwide. And the disparities among these diseases are massive. So for example, the asthma prevalence in the Global Biobank meta-analysis study that we have launched can vary anywhere from 2% in some biobanks to 20% in other biobanks. So there's an order of magnitude difference in prevalence depending on where you're looking. And within groups, we can also see huge prevalence differences depending on where groups are from. So for example, Puerto Rico has a much higher prevalence of asthma compared to, for example, Mexican Americans. And clearly this is not driven by ancestry differences, but is clearly much more likely driven from an exposure side. So we're really interested in trying to leverage genetic information as well as non-genetic information to try to understand why these are comorbid but vary so much across populations. So Yixuan He is a postdoc in the group who was looking at the shared genetic risk factors among respiratory traits. So for example, she looked at GWAS of asthma, COPD, lung cancer. Many of these, well, COPD and lung cancer have a high comorbidity with smoking, asthma in children, not so much. These also are highly correlated with spirometry. So basically breathe into a tube as hard as you can for a second or at full capacity. That's kind of what spirometry is measuring, your overall lung capacity. We looked at how genetically correlated these were and saw very high correlations as expected based on their comorbidities. And so we saw a potential here to leverage the relationships between these traits to improve genetic discovery, but also pull apart what those mechanistic pathways were along the way. There's a huge amount of heterogeneity even within these sort of subclasses of disease. So for example, there's eosinophilic versus neutrophilic asthma has kind of been described in the literature, similar for COPD. And it's clear that some biological drugs work a little bit better for treating some of these subtypes compared to others. So we wanted to understand, is the genetics helping underpin and explain some of these pathways and why this heterogeneity exists? So to do this, there's just a complicated kind of discovery and study design setup where we ran the largest multi-trait and multi-ancestry GWAS and combined we ran some GWAS for spirometry and then leveraged some existing data that was out there and combined all of this in a multi-trait and multi-ancestry way. So one of the first and coolest things that I want to just point out from this study is that we found shared and distinct etiology among lung diseases and smoking. So smoking is a huge known risk factor for both COPD on the left part of this plot, as well as lung cancer on the right part of this plot. That is very, very clear. That's been well characterized by all epidemiology. But what we're interested in is, are there genetic risk factors that are clearly related to these diseases, but do not necessarily relate to smoking risk? So you can see in the blue genetic variants that seem to be relating to COPD and lung cancer because they are influencing smoking risk. And then in the yellow are related to these diseases, but not through a smoking related pathway. We see some cool validated hits here. So for example, for lung cancer, we see a BRCA2 mutation as well as a TERT mutation. BRCA2 is well characterized for its breast cancer links, but it also relates to lung cancer risk. For COPD, we also see some well-characterized associations, CFTR, the cause of cystic fibrosis, as well as SERPINA1, which is basically a phenocopy of genetic emphysema. But there's a lot of new things also to try to understand why some populations, for example, non-smoking individuals who are diagnosed with COPD may have a predisposition towards developing those diseases. She also developed a polygenic risk score based on the analysis relating all of these genetically correlated traits. And in general, in the green, what I'm going to show you, can you see my mouse? Yeah. In the green is showing a baseline model of trying to predict COPD on the left, asthma on the left versus COPD on the right. So age and sex, we're starting with, let me just summarize the results. The results here are PRS Extra improves our prediction of asthma and COPD. It doesn't do that much better than the standard PRS, but leveraging all of this information helps a lot. Where we saw the biggest gains were in admixed American populations with the known largest degree of heterogeneity. And we think the reason for this is because we've conducted a large study in East Asian ancestry populations for spirometry, which has the closest genetic relationship with these admixed American groups. Just genetic divergence wise. And so this is likely disproportionately improving our prediction accuracy in these groups. Okay, I just want to end on one new research area that we're kind of exploring related to this respiratory disease space. We've been really excited by all of the progress in proteomics data. The reason for that is that protein scores have really high predictive value for incident disease. So basically you draw someone's blood, you follow them for some time, you see if they develop a disease, and you ask how predictive at that original baseline measure those proteins were at predicting that disease. Overall, we see that these proteins have really strong predictive utility for things like death, I guess the ultimate disease, as well as respiratory diseases like lung cancer and COPD. So that's what you can see in the green. If we add on lifestyle risk factors, so things like BMI and alcohol use and other measures, including smoking, that's what's shown in the blue kind of comparison expanded baseline model versus what happens with a protein score added on top of that. And this prediction model really attenuates when you add on those lifestyle risk models, indicating that these protein profiles are really dynamically measuring some causal risk factors that are genetically mediated, but also a huge amount of our lifestyle. These are really dynamic biomarkers. They're really responsive to our social and environmental exposures as well. So we really wanted to dig into this for these lung diseases in particular, since we were already kind of focusing on this. I'm going to skip a little bit for the sake of time and just mention only 8% of these incident disease associations have some Mendelian randomization evidence. That is to say, only 8% of proteins that are predictive of incident disease seem to have some causal underlying basis driven by genetics. However, they are really, really predictive of disease. And when we look at how well various lifestyle and risk factor and other environmental risk factors are at predicting these diseases, a huge amount of that is coming from things like smoking. If we look at how predictive these non-Mendelian randomization evidence proteins are at predicting a variety of different diseases, we also see that they're much more pervasive. So Mendelian randomization causal evidence proteins seem to be fairly disease specific, versus if you're looking at how predictive a bunch of different proteins are at disease, they're doing a good job at predicting disease, but not just one disease, they're predicting many different diseases. So that's something worth kind of keeping in mind as we move forward in the proteomic space. Kristen built a smoking protein score. Kristen's a fantastic PhD student I mentioned before. She's co-advised with Mark Daly. We use data from the UK Biobank protein pharma partnership or something like that, using about half of the sample for training and half for testing. We trained just using never smokers versus current smoking status. And then we used Lasso to build this predictive model with tenfold cross-validation. This model picked up on hundreds of proteins and yielded an AUC for smoking status of 0.95, which is an AUC I don't think I'm ever going to see again. So it is incredibly accurate at predicting these things. The vast majority of proteins that associate with incident disease are also smoking associated. So overall, when we looked at the plasma proteome, we saw that about 66% of proteins that were predictive of disease were predictive of smoking as well. Just for the sake of time, I just wanted to mention these smoking protein scores really stratify smoking status, but they're really biologically dynamic. So they also stratify time since smoking in previous smokers, and then they stratify pack years in current smokers. So they are kind of capturing like the biological smoking accumulation over the life course as opposed to just what is your recent smoking status. Okay, so I want to end with some key takeaways and conclusions to allow time for a couple of questions. I'm going to skip to my key takeaways slide and just mention diverse genetic studies are the best for everybody. I'm really excited by the potential for pleiotropy dissection analysis across multiple traits to give us some well-powered mechanistic insights into different pathways that are helping inform our understanding of disease biology. Genetics provides a uniquely powerful tool for understanding heritable traits and diseases, as well as different genetic architectures. They're coming for precision medicine, and they are likely to improve clinical models, and that is very exciting. But currently, they're also likely to increase disparities due to vast Eurocentric study and other biases. Molecular biomarkers like proteomic profiles, for example, are also coming to precision medicine, which is very exciting. But it's important to delineate and understand what's the causal basis of these biomarkers and what's poorly understood and maybe needs a little bit more refining. With that, I just want to say thank you so much for coming and for listening. If you're sick of perfect weather and you're thinking about some next steps, come talk to me. Thank you all so much, and I'm happy to take any questions. Yeah. Yeah, that's a great question. So in the Pan-UK Biobank project, for example, we had vastly imbalanced sample sizes. When we're thinking across UK Biobank and all of us with different ancestries, how we should run our statistical models, one thing that we did was compared like a meta-analysis approach versus a mega-analysis approach. And there's a lot of literature out there saying like a mega-analysis approach should be a little bit more powerful, but a meta-analysis might be a little more convenient if you're doing a federated analysis. We weren't doing that. But in our hands, if we tried to mega analyze everybody, so that is put everybody all in the same statistical model, account for genetic relatedness and for ancestry at the global level, as one is like commonly doing in GWAS, we saw hugely inflated type 1 error for a subset of traits with the mega analysis approach and had to partition out into ancestry groups to appropriately control for our type 1 error. So it depends on the data set how you need to like group those individuals to control that type 1 error, but that's what we've seen generally. Yeah. Great question. Not at all. I'm sure some of my colleagues have, but I have not. Thank you. Sorry. Yeah. That's a good question. I think the pushback we get more from clinicians tends to be on like phenotype definition as opposed to how many diverse data sources we're using. There's often a push for really refined phenotypes that we just don't have the ability to look at with that granularity at scale. In terms of these multi-omic factors, there's some really exciting, cheaper types of biomarkers that are coming. So for example, metabolomic data is coming easily available to large-scale biobanks. You can measure hundreds of metabolites for like $20 a sample, which is getting you down to like complete blood cell count, routine lab tests. I think the lab tests, if you're starting to add up whole genome sequencing with proteomic profiles, which are hundreds of dollars a sample, with metabolomics data, that becomes very cost prohibitive very quickly. So I think you're right to have your finger on that pulse, but that's not pushback we've gotten so far because I think we're still in, basic science land as opposed to translational. Yeah, Tiffany.
So I wanted to ask, like, are your future plans for that this development methods, and is there an opportunity to try home trade?
I love that direction. And I think it's probably got some promise, especially because I've seen multi-ancestry fine mapping methods kind of exploding in the last year. There's been a bunch of them, but I haven't actually seen so many multi-trait takes on that. But because of the genetic correlation, I bet there could be some promise, especially because like we spend a lot of our time with multi-trait data kind of staring at like this association only shows up in schizophrenia, but this one seems to be across psychiatric diagnoses. So if there was a way to easily kind of summarize this pops up here and not there, I think that'd be really great. Yeah. So there is a lot of work in that space. We have not led that work. Others have. So for example, there's a so-called female protective effect for autism spectrum disorders that's been well researched. I think the jury's still out on what the contributions necessarily from genetics versus cultural versus other factors to that are. In the studies that we're looking at, primarily focused on psychosis, schizophrenia, bipolar disorder, we've taken a less fine scale look at, sex differences, partially because many of them seem to be relatively equal opportunity, unfortunately, and also related to, especially genetics, but also other risk factors like substance uses and other things as well. But it's a great topic, just not one that we've focused on.